Glycogen Metabolism, Gluconeogenesis, and Integration by Laureano Andrade Vicente and the MCAT Disciples. So here are the AMC topic list of what we're going to go through. We've already gone through all of these things, so I've crossed them out, except for the pentose phosphate pathway. I haven't gotten there yet. But we're going to do the rest of these, and let's get right on to it. Introduction to the players. So here's PLP, or pyridoxal phosphate. All right, it is a relatively ubiquitous cofactor that's used in transaminases and glycogen phosphorylase. The reason I'm mentioning it, of course, is it's being used in glycogen phosphorylase. Turns out that PLP is actually derived from vitamin B6, the pyridoxal group of vitamins. And the reason why I mention that is because the MCAT wants you to know that B vitamins play a critical role as cofactors for many different biological reactions, and here's just one of the examples. So moving forward, you're going to see this portion of the molecule actually working the transaminase reaction mechanisms. Of course, I'll never show you that, but just for you to be aware of. But in terms of glycogen phosphorylase, where we're going to see PLP working, and you'll see this later on, is this phosphate group here. It's actually going to be working in an acid-base catalysis mechanism. And so you'll be seeing that in the mechanism of glycogen phosphorylase, to break down of glycogen into free glucose. Anyway, that's PLP. Moving on to UTP, it's just uridine triphosphate, right? It's the same thing as ATP in the sense that it's just an NTP molecule, but it's just uracil based instead of adenosine. But everything else has the same exact properties in terms of energy. And again, it's energy equivalent to ATP. You can write an equilibrium reaction with UTP and ADP to form then ATP and UDP. So there's nothing magic about it. It's just uracil. And it's just built, the enzyme is built to use UTP instead of ATP. That's all. So that's it. We can move on then to glycogen. So glycogen is a storage form of glucose. It's just a bunch of glucose residues connected together via alpha-1,4 and alpha-1,6 linkages. These alpha-1,4 linkages are found within the straight chains of glycogen, and they are branched at alpha-1,6 positions. Alpha-1,6 because it's at the 1 position here and the 6 carbon here. So alpha-1,6 linkage. And it's alpha. Remember that nomenclature. Alpha means that it's going down relative to the hydrogen that's going up, and beta means it's going up relative to the hydrogen that's going down, right? And it had nothing to do with this guy right here. This is actually D-glucose, right? Because it's going up, and if this was going down, it'd be L-glucose. But again, glycogen is just a storage form of glucose. Now, you'll read about starch. So starch is what plants make carbohydrates. Starch is the storage form for plants of glucose. And the only thing different about starch and glycogen is the way that the structure of it, the way that it packs. Because it still has alpha-1,4 linkages and branches at alpha-1,6 linkages. It's just the way that it packs. So objectively, there's nothing really different about starch and glycogen. It's just the fact that we don't make starch, we make glycogen. And the only thing different about it is the way that it packs and the structure of it. How many branches it has and where does it have branches. Glycogen is a storage form of glucose. Glucose residues. Glycogen is found primarily in liver and muscle tissue. Now, in the liver, it plays a very important role in regulating blood glucose levels. We want to maintain a dynamic, steady state of about 5 millimolar, or 100 milligrams per deciliter of glucose, okay? We want to maintain that. And what's a dynamic, steady state? Well, it's a state at which the forward reaction equals the reverse reaction, which is just like an equilibrium state, but the only difference is that we need to put in energy to maintain that state. We need to run, we have to run a bunch of reactions and use enzyme catalysis to maintain that steady state. Where at that equilibrium, that is a natural state, a natural process, entropy is maximized, and the rate of the forward reaction is equal to the rate of the reverse reaction. But our dynamic steady state requires that we put in energy to maintain that state. So dynamic, right? We have to put in energy to maintain that state. On a net basis, the state isn't changing, but of course, the forward reaction and reverse reaction are still happening, but they're just, the rate at which they're happening are equal to one another, so that the state is steady. All right, so we need to maintain 5 millimolar, dynamic steady state, and we use a bunch of hormones and allosteric regulators and covalent regulators to maintain that state. Now, we also find glycogen a lot in the muscle, and it uses it for short bursts of exercise. And while, the glycogen liver, the glycogen, we have much more glycogen in muscle than in liver, probably just due to the fact that we have much more muscle tissue than liver. But again, it's really important for liver because of the fact that we need to maintain this 5 millimolar level of blood glucose. Because again, if we ever drop below about 2.5 millimolar, we're going to go unconscious. Our brain needs that much glucose. Our brain is very keen on having its glucose, or at least having its ketone body. So if you don't have one or the other, you're going to have big problems with brain. Glycogen is found within the cytosol. All right, glycogenolysis, or glycogen breakdown, or glycogen synthesis, okay, or glycogenesis occurred within the cytosol. It occurs within the cytosol. You'll notice it's protein glycogen. We'll talk about that. But it occurs within the cytosol. Remember that. Another thing that I want to mention it now, is that both glycogen synthesis and glycogenolysis occur within the cytosol. So clearly, we don't want those happening at the same time. Otherwise, we'll just be running a futile cycle where we just waste ATP building up the glycogen, then break it down again to build it up again. And so clearly there are allosteric regulators here that are very potent and have play a big role in preventing that vicious cycle, that vicious futile cycle. All right, so we'll move then into glycogen synthesis, or glycogenesis. That's the terminology. So here's the process diagram. We're doing glycogenesis, so why don't we start at glucose-6-phosphate, and we'll go through a broad stroke, and then we'll break it down one by one. So phosphoglucomutase will convert glucose-6-phosphate that we get from glycolysis. The first step in glycolysis, remember hexokinase or glucokinase in the liver to form then glucose-6-phosphate. We can convert that into glucose-1-phosphate via an isomerase type enzyme. Okay, a phosphoglucomutase, right? That's the mutase of the isomerase, gives free energy about 0. And it uses a reaction mechanism very similar to the phosphoglycerate mutase. But the only difference here is that it uses a serine residue as opposed to a histidine residue to transfer the phosphate group. So you'll see that. But we get glucose-1-phosphate. Then we use UDP glucose phosphorylase to activate that glucose to form UDP glucose. All right. And the way it does that is just a simple nucleophilic attack. The glucose-1-phosphate phosphate group attacks the alpha phosphate group and causes pyrophosphate to leave. Now we get UDP glucose. And UDP glucose via glycogen synthase will form then glycogen. We'll add on to the glycogen polymer. Now, that again, UDP glucose is the high energy molecule, so you don't need any input of ATP, and that's why it's called a synthase. There's no NTP input here. And you're saying, well, you put an NTP here. Well, no, that's earlier. That's a different reaction. So now we have glycogen. So we'll go through that. Let's go through it. Again, glucose-6-phosphate can go through a number of different pathways. It can go through glycogenesis to form glycogen. It can go back via gluconeogenesis to form glucose. It can go via the pentose shunt to form ribose 5-phosphate and NADPH. Or you can put it through glycolysis to form acetyl-CoA and eventually phospholipids, fatty acids, cholesterol, ketones. But what we are kind of doing here is glycogenesis. So we want to put it through glycogenesis. So let's do that. Again, phosphoglucomutase will convert G6P into glucose-1-phosphate. It's a reversible reaction. You know, just remember, a gives free energy of a 0 indicates that it is at equilibrium. Remember that. Remember, because then delta G standard equals negative ln KQ. Anyway, the mechanism here, you have the serine residue with the phosphate group that had been primed with an ATP molecule. You have this nucleophilic attack by this OH, okay, and that transfers the phosphate here. And then you get another nucleophilic attack by this OH on this phosphate group. And then you end up getting glucose-1-phosphate. Simple reaction. Nothing special there. And that is a conversion of glucose-6-phosphate to glucose-1-phosphate. All right, so we got this far. Now we want to convert that into UDP glucose. But before we do that, before we go through that process, we need to talk about glycogen. Glycogen synthase, the enzyme in question, require the primer before it can polymerize glucose into glycogen. Glycogenin is the enzyme that catalyzes the formation of that primer. Glycogen is very similar to, let's say, Pol alpha primase if you're talking about DNA replication. Okay, glycogenin acts as a glycosyl or glucosyl transferase to transfer glucose molecules to then form a glycogen primer. So again, a glycosyl transferase transfers sugar phosphate moieties or molecules. If you're talking about glucose transferring, then it would be a more specific nomenclature would be a glucosyl transferase. If you're talking about fructose, you would call it a fructosyl transferase. That's the nomenclature. And again, what glycogen does, it is a glucosyl transferase, and it transfers glucose molecules to form this primer of glucose molecules, so that then glycogen synthase can then catalyze the reaction. It needs that primer. Okay, its active site recognizes at least 4 residues before it can add a residue at the end. It serves as a primer for extension, okay? And then glycogen synthase, again, it recognizes these four and then adds one on at the end here, okay? So that's the process. It needs to recognize these. Its active site needs to recognize these, or part of the enzyme needs to recognize these to then add a glucose residue here. A residue is just what remained of a glucose molecule, by the way. And notice it's an alpha-1,4 linkage. That's important. Enzymes are very specific, guys. The active sites are very specific. There are the difference between the 1 position and the 4 position and the 1 and the 6 position. Clearly, this is not the same structure as this. So clearly, the active sites are going to be built to form either alpha-1,4 or alpha-1,2 or alpha-1,6. But it's going to be specific to that kind of linkage because most active sites are very specific to what it's going to bind to. So now we're at UDP glucose. I mean G1P to form UDP glucose to then go through glycogen synthase. Alright, so we need to form charged UDP glucose. So charged glucose is just a high energy form of glucose. And what we need to do there, we take glucose 1-phosphate, we do nucleophilic attack on the alpha phosphate group of this UTP molecule, and we cause pyrophosphate to leave. Pyrophosphate leaves, and we can further break that down via the very ubiquitous, the omnipresent inorganic pyrophosphatase to form 2 inorganic phosphates, and we have the UDP glucose. Okay, and it's forming UDP glucose because it already had a phosphate here, so it just added another one. And that reaction is actually the formation of UDP glucose is actually relatively unfavorable, but it's catalyzed by this breakdown of pyrophosphate into 2 inorganic phosphates to form an overall negative delta G value. And recall, I'm just going to remind you about this now. You cannot catalyze a reaction that is unfavorable. You will never make a reaction go forward if it's an unfavorable reaction unless you put in energy. But by an enzyme alone, you will never cause an unfavorable reaction to occur by its own. It cannot make an unspontaneous reaction spontaneous. All it does is increase the reaction rate of the spontaneous reaction. Okay? Keep that in mind. UDP glucose now is a high energy molecule, can be used to then form glycogen via glycogen synthase. So glycogen synthase catalyzes the formation of alpha-1,4 glycosidic bonds. It has the general classification of a transferase. It is under allosteric regulation. Glucose-6-phosphate will activate it. Again, but alpha-1,4 glycosidic bonds, not alpha-1,6, alpha-1,4. This is the bond that it can catalyze and form. Again, that's just due to the active site and the way the enzyme is built. We'll only recognize the alpha-1,4 position because those are the positions where it just fits within the active site and all the amino acids recognize, all the moieties that it needs to recognize. So you take UDP glucose. You form an oxonium-ion intermediate. How do you do that? Well, what happened is just the UDP left. UDP acted as a leaving group. It was stabilized by glycogen synthase and it left. There were probably some lysine residues that were interacting with these UDP phosphate groups and caused it to become a better leaving group and it left. So now we have an oxonium-ion intermediate. This intermediate is a so-called half-chair conformation, okay? In that half-chair conformation, what happens is you get a nucleophilic attack by the glycogen OH group at the 4-carbon position. And then it attacks. Look at that. You form glycogen. You added 1 glucose molecule to your glycogen residue that originally had 3, now it has 4. And again, it's an alpha-1,4 linkage. Nothing difficult here. One thing to notice, actually, we'll wait. Let's not. Alpha-1,4 glycosidic bond. Again, this is the glycosidic bond. It's just an ether bond. It's just a bond between 2 carbohydrates. Now we have that. We have glycogen. We made glycogen. We made it longer. We're storing our glucose that we ate from our meal. Right? This would be happening when we ate a meal, right? And now we want to branch it a little bit. So we want to form branches. So what happens is our glycogen synthase just keeps making these really, really long branches. And the reality of it is, as you'll come to appreciate when we go through glycogenolysis, the breakdown of glycogen, it's very important for us to have non-reducing ends. Because if we don't have non-reducing ends, we're going to run into big trouble in terms of breaking down glycogen to get it very quickly into then the muscle cytosol to then use it through glycolysis to generate ATP and cellular respiration. So we want to make branches so we have more non-reducing ends, right? This is a non-reducing end. This is a non-reducing end. We want to create more of those. So we're going to move this right here. Okay, this alpha-1,4 linkage. We're going to cleave it and then reform an alpha-1,6 bond via glycogen branching enzyme. And that's what we did. And you'll notice that those branches appear about 8 residues down, 8 to 12 residues down from another branch. So you don't get branching, let's say, from this point, from like a branch here and then a branch here. No. It has to be at least 8 to 12 residues away from another branch. And that just results from packing issues. Okay, packing issues. You want the branches to be a little bit more spread out. But again, glycogen branching enzyme catalyzes the formation of alpha-1,6 bonds and breaking of alpha-1,4 bonds. Nothing crazy there. There it is. Alright, glycogen structure results from the need to pack plus need for quick access. The deep branching enzyme, which we haven't talked about, we just talked about branching enzyme, by the way. I mentioned deep branching enzyme because it's important to talk about structure. So deep branching enzyme is slow. It's much slower than glycogen phosphorylase, which is the breakdown enzyme. But we haven't talked about that yet either. I don't know, but for me, when I look at glycogen, I would say, why would we want branches? Wouldn't that make it harder to pack glycogen? And that's true. But the reality of it is we want more non-reducing ends so that we can remove the glucose residues faster. We want that because of the fact that glycogen phosphorylase is much faster than the deep branching enzyme. So the more non-reducing ends we have, the more the quicker we can release the glucose into the cytosol. So it's a balancing act between the need to store glycogen and the quick release of it. And it results in branches every 8 to 12 glucose residues. That's just what nature has chosen for glycogen. And starch is different. Starch has branches and I don't know how many residues, but it's different. It has a different structure, overall structure. Again, glycogen synthase can be allosterically modulated. Glucose-6-phosphate will activate glycogen synthase. And think about why that is. If you have a lot of glucose-6-phosphate, clearly then you have a lot of glucose. If you have a lot of glucose and you're saturated with glucose, what you want to do is then convert it into glycogen. By mass action, it's going to push the formation of glycogen, right? Because then we get rid of glucose and we return back to our steady state, 5 millimolar. We want to maintain that dynamic steady state. Anything that's going to be against that dynamic steady state, we're going to move in ways to go back to that steady state. So glucose-6-phosphate will activate glycogen synthase. All right, that was synthesis. Let's talk about breakdown now. Glycogenolysis. Here we are. We have glycogen that we just made, and now let's say we are starving and we need to increase blood glucose levels. All right, or we're exercising. We need to break down glycogen. Well, the first step, really, both of these. So these things are really happening in tandem in terms of debranching and glycogen phosphorylase, but we're breaking it up superficially. Other thing to know about it, it has many different active sites. It is two to be particular. Well, two to be particular, because what it does is it breaks down in one active site. It breaks down alpha-1,4 bonds like this one right here, and it will form the alpha-1,4 bond right here again in that first active site. So let's see that. What it did again is it cleaved this and it put it right here. Okay, it's debranching. It's adding on to the end of the glycogen tail. And then the next step then is to break this alpha-1,6 bond. So it has to use a different active site for that, because again, I told you, if you have an active site recognizing an alpha-1,4 bond, it's not recognizing an alpha-1,6 bond. Okay? It's a specific interaction. If you don't, you're not going to fit both of those in there at the same time. So you're going to have to use a different active site, same enzyme, different active site, to break that alpha-1,6 bond. One thing you'll notice is that it didn't add on at the end. If you count up these, you'll notice that it didn't add on at the end. It didn't. 10% of glycogen is released as free glucose as opposed to glucose 1-phosphate. So meaningful to you is just to say that, know that fact, that glycogen debranching and then will release some as free glucose. So now we can talk about glycogen phosphorylase from glucose 1-phosphate. So we take glycogen phosphorylase, and we form glucose 1-phosphate and glycogen. Okay? -1, right? We lost one of the glucose molecules. Okay, we released one. It's a very fast reaction relative to glycogen debranching enzyme. Okay, and again, you have to put an inorganic phosphate because it's a phosphorylase. You're adding a phosphate group. Okay, and you're cleaving some molecule. So here's the mechanism. Here's that PLP that I mentioned earlier. I'm not showing the full structure. I'm just writing PL, then the phosphate group that comes off of it. What happened is it acts as an acid. It acts in acid-base catalysis. It acts as an acid to protonate this phosphate group that can then protonate the glycogen molecule, so that one glucose molecule can leave, which it does. So now we have glycogen N minus 1, and now we have to deal with its oxonium ion intermediate. Again, notice the trend here. You see an oxonium ion in both glycogen phosphorylase and glycogen synthase as an intermediate. Now what happens is the free phosphate group that we added then attacks this oxonium ion to form glucose 1-phosphate, and we reform our enzyme by taking up that proton again that we had originally donated. Now we're back to glucose 1-phosphate. Now we add glucose 1-phosphate and glycogen N minus 1. Nothing crazy, nothing special. We're at glucose 1-phosphate again. So now we have to go through phosphoglucomutase to form glucose 6-phosphate. And again, this is that equilibrium. So this is controlled by mass action. This is controlled by Le Chatelier's principle. If you increase the amount of glucose 1-phosphate you have in cytosol, you're going to push this reaction to the left. Okay? You're going to push this reaction to the left. You're going to form more glucose 6-phosphate that will then, by mass action, push glycolysis or go out into cytosol or wherever it needs to go. Again, there's many fates for glucose 6-phosphate. It can go through glycogenesis to form glycogen, which obviously it wouldn't do if it just went through glycogenolysis. It can go through gluconeogenesis from glucose, pentose shunt, ribose 5-phosphate, form acetyl-CoA via glycolysis, pyruvate dehydrogenase, and electron transport chain, TCA cycle. Okay, we can go many different ways with this. Alright, glycogen phosphorylase can be allosterically regulated. So it has a T state and an R state very similar to what hemoglobin does in terms of T state. Remember, in hemoglobin, T state is going to be the deoxygenated state, and R state is going to be the oxygenated state by and large. It's going to be stabilized by oxygen. In this case, the T state for glycogen phosphorylase is less active. R state, it's more active. That's the terminology. It gets kind of confusing, but the terminology is used kind of loosely. The T state less active, R state more active. We're not saying active or inactive. We're just saying less active and more active. Some allosteric regulators. ATP binds to and stabilizes the T state. G6P binds to and stabilizes the T state. Glucose at the liver binds to and stabilizes the T state. Again, this should intuitively make sense to you. You don't want, especially in liver, if you have high levels of glucose, high levels of ATP, that just means that you're in a high energy state. You don't need to be breaking down glycogen stores if you're in a high energy state, if you have a lot of glucose, right? So these allosteric effectors will bind to and stabilize the T state. R state is more active, and AMP will stabilize the R state. Again, AMP, big regulator here. AMP will bind to this protein here and cause the allosteric shift to the R state, causing it to be more active, causing for the breakdown of glycogen, okay? So that you can then have glucose 6-phosphate, to then glucose 1-phosphate, then glucose 6-phosphate, to then go through glycolysis and citric acid cycle, electron transport chain, to then form ATP, to get rid of this AMP, high levels of AMP. But there's another piece to this now in terms of regulation. It's the hormonal piece, but there's also the piece that happens in muscle tissue. So in muscle, calcium ion helps regulate glycogen metabolism, particularly phosphorylase, glycogen phosphorylase. What happens is that, as you know, when you contract muscle, there's calcium release from the sarcoplasmic reticulum. Okay, that calcium is bent on binding troponin, causing conformation chain in troponin tropomyosin complex, revealing the myosin binding site on the actin filament. Myosin head binds, goes through cross cycling. Okay, power stroke, releases ATP and organic phosphate. ATP binds, and now you are doing cross-bridge cycling. But that calcium can also activate phosphorylase kinase. And then that phosphorylase kinase will phosphorylate glycogen phosphorylase. And then that will then activate glycogen phosphorylase to then break down glycogen into glucose to then provide carbohydrates, okay, glucose, for glycolysis, citric acid cycle, and electron transport chain to generate the ATP required to do muscle contraction. The other thing that is significant here is, and that's why it makes sense. Okay, so calcium ion helps regulate glycogen metabolism in muscle because of the fact that muscle tissue, whenever you have calcium, you need calcium to get contraction. So whenever you have calcium, you're going to be using up ATP in muscle cells. So you better replace that somehow. That is how we regulate that in muscle. So here's a question for you. You run an experiment to test the efficacy of an inhibitor. You put the inhibitor at very high concentration in a tube containing either glycogen synthase or glycogen phosphorylase under physiological conditions. you find that in both tubes, the inhibitor is very effective. You theorize that A, it is most likely a competitive inhibitor since both enzymes have similar intermediates. B, it is most likely a non-competitive inhibitor since the enzymes catalyze the reverse reaction of one another. Or C, it is most likely uncompetitive inhibition because the Km and the Vmax decrease proportionally. Or D is most likely mixed inhibition because the inhibitor can bind to both enzymes. What's the answer? Turns out it is A. The answer is A. And it is a competitive inhibitor, and the reason why I say that is because the inhibitor is effective in both solutions. And remember, the oxonium-ion intermediate is found within both glycogen. The oxonium-ion intermediate is found within glycogen phosphorylase and glycogen synthase. So a competitive inhibitor would inhibit both of those very effectively. So 1,5-gluconolactone is an example of an inhibitor of glycogen phosphorylase and glycogen synthase. So let me review with you very quickly the different types of mechanisms of inhibition. So competitive inhibitors bind to the active site. They make the Km appear larger than it is, but they don't change the Vmax. They don't change the Vmax because you can saturate the enzyme and outcompete the competitive inhibitor. But they increase the parent Km because you're pulling enzyme out of solution. So you have to put in more substrate than measure a Km. You have uncompetitive inhibition. So uncompetitive inhibitors bind to the enzyme substrate complex, and they pull the enzyme substrate complex out of solution and distort the active site. And what the effect of that is that you decrease the apparent Km. The apparent Km decreases. Apparent Km now. The apparent Km decreases because you're pulling enzyme substrate out of solution, so it's easier to form than the enzyme substrate complex. Mass action. But the Vmax also decreases. The Vmax also decreases such that the slope of the Km or Vmax stay the same. Okay, so the Vmax and the Km decrease with uncompetitive inhibitors. Non-competitive inhibitors, there is no change in the Km, but the Vmax decreases. Okay, and the non-competitive inhibitors allosterically distort both the enzyme substrate complex as well as the enzyme alone. Okay, and then there's mixed inhibition. Non-competitive inhibition is just a form of mixed inhibition. And mixed inhibition does the same thing non-competitive does, but the apparent Km can either increase or decrease, but the Vmax will always decrease because it's an inhibitor. And those are the different types of mechanisms you can look at. So we'll go through some tissue-specific glycogen metabolism events that occur pretty much after glycogen metabolism. The actual regulation and the actual glycogen metabolism happens pretty normally in the tissue where glycogen is found, liver and muscle. But in the function of glycogenolysis in liver is to regulate blood glucose levels. So what happens is we break down glycogen via glycogenolysis into glucose-6-phosphate. Well, glucose-1-phosphate, then glucose-6-phosphate via phosphoglucomutase. And then what happens is it's all functioning to regulate blood glucose levels, so we need to convert a glucose-6-phosphate into glucose to then send that out into the periphery. So how we do that is a glucose-6-phosphatase that is found within the lumen of the ER, which is then converting glucose-6-phosphate into glucose, which is then transported out into the cytosol, which can then go out via GLUT2 transported into bloodstream. Well, extracellular fluid space and bloodstream. Now, the significance of this is that you only find this glucose-6-phosphatase in liver. You don't find it in muscle, so muscle is never going to push out glucose into bloodstream. Now, this is only found within liver, glucose-6-phosphatase. Now, the other thing that's important here is that GLUT2 is not found in muscle, and it's not under insulin regulation. So liver is going to put out glucose regardless of whether the insulin is present or not. These GLUT2 transporters are found within liver hepatocytes, okay? And it's not under insulin regulation. So they're going to put out glucose no matter what, if they need to. Liver functions to regulate blood glucose. The other part of this, which is relatively important, is the fact that within the glycolysis that occurs within the liver, the enzyme that catalyzes that first step, glucose to glucose-6-phosphate, is glucokinase. Glucokinase has a very high Km. It has a very low, not very high, but a high enough Km such that it has a rather low binding affinity for glucose. So if glucose levels are low, then in blood rather, then glucose is going to flow out down this concentration gradient into blood, and it's not going to be bound by glucokinase. If glucose levels are high, then glucose will be bound by glucokinase to a higher extent, and less will go out into blood because of the fact now you have a concentration gradient in, and then it can go into glucokinase. Whereas, as you'll see in muscle cell, muscles prolifically break down glucose. Okay, the Km for hexokinase is very low. The Km is about what? 1 millimolar. So to reach 1/2 Vmax. So we're at Vmax for hexokinase at, well, right around 5 millimolars of glucose. So any glucose that gets to muscle, either via a glucose transporter, a GLUT4 transporter, or via glycogenolysis, it's going to go right into glycolysis. Okay, it's going to go right into glycolysis. Muscle is going to break down glucose. Muscle does not contribute to blood glucose level. It will never shoot glucose back into blood. It's always going to go through glycolysis. So here are the different GLUT transporters and their distribution. I'm mentioning this because it kind of gets confusing if all you ever talk about is GLUT4 and GLUT2. So GLUT1 is found in the blood-brain barrier and endothelial cells and the erythrocytes and all cells at a basal level. So at basal low level, all cells express GLUT1. Now GLUT2 is found in the renal tubular cells. Okay, so the proximal convoluted tubule, distal convoluted tubule, collecting duct, pancreatic beta cell, and liver cells. Okay, pancreatic beta cells. GLUT3 then is found in neurons. Okay, again, notice that neurons don't give a hoot. Blood-brain barrier doesn't give a hoot about insulin, because insulin only regulates the level of GLUT4 transporters in muscle and adipose tissue. So the brain doesn't give a hoot about how much insulin is in blood. It just cares about getting enough glucose and having enough glucose in blood. So if you have too much insulin, say you inject too much insulin, you become hypoglycemic. You go into hypoglycemic shock. It's not that brain cares about how much insulin is in bloodstream. It just cares about how much glucose is in blood. And if you inject too much insulin into you, you're going to remove a mass amount of glucose from blood and into the cells. And then you go into hypoglycemic shock. So the treatment for that, and it's counterintuitive, but the treatment for that is to give the diabetic person some sugar. Because the point is to keep yourself from falling out of consciousness. And keep yourself from brain from being damaged. How you do that is provide glucose. All right, glycogen storage diseases are genetic disorders that negatively impact glycogen metabolism, producing glycogen that is abnormal in either quality or quantity. Okay, that's a glycogen storage disease. Now, there's a bunch of different types. I'll go through a few of them. There's von Gerk's disease. It's type 1. And it's just a glucose-6-phosphatase deficiency. A glucose-6-phosphatase deficiency. That enzyme in liver and in some points kidney that catalyzes the breakdown of glucose-6-phosphate into glucose so that it can then be transported out via the GLUT2 transporters. Well, that's deficient in von Gerk's disease. And that's going to cause you big problems. It's going to cause you a large accumulation of glycogen in liver and kidney. Why? Why? Because you would think then that if you have a glucose-6-phosphate buildup, then it can just go through glycolysis. And yeah, it can. But glucose-6-phosphate is an allosteric activator of what? Glycogen synthase. So you're going to have a buildup of glycogen in liver plus kidney. Okay, because that glucose-6-phosphate is going to activate glycogen synthase to form more glycogen. And that leads to hepatomegaly and hypoglycemia. Hypoglycemia because of the fact that now the liver cannot push out glucose into bloodstream or kidney. Can't push glucose out into bloodstream because of the fact it doesn't have glucose-6-phosphatase. So you get hypoglycemia. You can't really regulate. Can't keep that dynamic steady state of 5 millimolar. Another type, McArdle's disease, type 5. It's a glycogen phosphorylase muscle enzyme isoenzyme deficiency. So an isozyme is just an enzyme that is very similar to another enzyme, but it's just slightly different. So it's an isozyme. Just like an isomer is a very similar compound to another compound. And it leads to muscle cramps and pain upon exertion. Why do you think it leads to muscle cramps? Well, I told you earlier that calcium and muscle will activate phosphorylase kinase to activate glycogen phosphorylase. Well, if you don't have glycogen phosphorylase, you're not going to be getting a lot of glucose to then power muscle contraction. You get muscle cramps. And the other part of it that is relatively important is to appreciate the fact that when you go through your power stroke, right, you release ADP and inorganic phosphate, but you will be stuck in that position. It's called the rigor state. You will be stuck in that rigor state until ATP binds. Well, if you don't have any ATP to bind because you don't have any glycogen phosphorylase to give you glucose, well, you're going to get muscle cramps. That rigor state is the same. That is where that rigor state derives from the fact that when we die, we get rigor mortis, right? We're stiff for a few hours after death. And that's due to the fact that you don't have any ATP to then release the myosin head from the actin filament. So that's where that's coming from. And that's HERS disease, type 6. It's a glycogen phosphorylase liver isozyme deficiency. Of course, then you're going to have hypoglycemia. Okay, so those are glycogen storage diseases. It's genetic disorders. Okay, you have some mutation, some polymorphism, or some deletion, or whatever, or some insertion that negatively impacts glycogen metabolism. So we'll move into now regulation of glycogen metabolism. Let's get right into it. The first thing we have to talk a little bit about a nomenclature, a weird nomenclature. So A and B. So you'll often see, let's say, enzyme A, and then you'll see it in enzyme B. So A, the active form, and B, the inactive form. Well, A and B do not indicate R or T states or phosphorylation states per se. Now, per se, they are related to one another, but per se, they don't indicate that. All it says is glycogen synthase A is the active form, and if you phosphorylate it, then it's glycogen synthase B. And that happens to be its inactive form. However, if you take glycogen phosphorylase and you phosphorylate it, it goes from glycogen phosphorylase B to glycogen phosphorylase A. Okay, but that is the active form. So it's not phosphorylation state per se. It is, but you can't say, well, if you're phosphorylated, you're B. If you're phosphorylated, you're A. No. A the active, B the inactive. So the way you remember that, you remember which one's which in terms of for which enzyme is, you got to think what makes the most physiological sense. Well, you know that glucagon induces phosphorylation signaling. I went through that in last lecture. It introduces phosphoryl kinase signaling. So it's going to phosphorylate things. Well, when glucose is gone, glucose is gone, you want to inactivate glycogen synthase and activate glycogen phosphorylase to increase blood glucose levels. So phosphorylation is going to lead to the inactivation of glycogen synthase and the activation of glycogen phosphorylase. Makes sense, okay? Same type of thing for insulin, except reverse terminology. So let's go through it. Glucagon or epinephrine. So I often say just glucagon, but it's epinephrine too, guys. I forget to include epinephrine in some things, but glucagon and epinephrine bind to the same kind of receptor that initiate the same kind of response, okay? So glucagon or epinephrine binding to the receptor will cause the conformational change in the G protein, causing for the activation of G alpha and the release of GTP and the binding of GTP. That'll cause dissociation of G alpha from G beta gamma. G alpha will move then and activate adenylate cyclase to convert ADP to cyclic AMP. Cyclic AMP will then activate protein kinase A. Protein kinase A will then activate, will phosphorylate phosphorylase kinase B into phosphorylase kinase A. It will also phosphorylate glycogen synthase A into glycogen synthase B. It will also phosphorylase kinase, remember this is the kinase we went through earlier, phosphorylase kinase will phosphorylate and activate glycogen phosphorylase. Alright, and this all makes sense, guys. Glycogen phosphorylase has been activated by phosphorylation, while glycogen synthase has been inactivated. This makes sense because of what we want to do. We want to bring up blood glucose levels. Okay, and that's the sequelae. That's the series of events there. And let's introduce to you some allosteric effectors now. ATP and G6 glucose-6-phosphate will bind to and inactivate glycogen phosphorylate, or stabilize the inactivation of glycogen phosphorylase. So stabilize the T state, and AMP will activate glycogen phosphorylase. It will stabilize the R state, or the active state. So one more, glucose in the liver only will activate glycogen phosphorylase, and glucose-6-phosphate will activate glycogen synthase A. So it'll stabilize the R state in glycogen synthase and activate it. Okay, so those are the allosteric regulators to be aware of. Remember, 1,5-glucono delta lactone will then activate both of these, competitive inhibitor. And now we can move on to insulin signaling. But so let's summarize. So glucagon or epinephrine will activate glycogen phosphorylase and then activate glycogen synthase. Now let's introduce insulin to the game. So this would be happening when we have high blood glucose levels. Now, insulin does a function of things. But glucagon, what happens when we do glucagon signaling is that PKA will then activate this phosphoprotein phosphatase inhibitor. So this is turned blue. And now it's inactivating phosphoprotein phosphatase. It's sequestering and inactivating it. However, when we get insulin signaling, what happens is that you activate phosphoprotein phosphatase. And you do that, in part, by dephosphorylating the phosphatase inhibitor. The point of the matter is PPP1, phosphoprotein phosphatase 1, is active when insulin is active, and what it'll do is reverse every single reaction that glucagon had affected via its cyclic AMP protein kinase A pathway. It will dephosphorylate glycogen synthase and dephosphorylate glycogen phosphorylase, and dephosphorylase phosphorylase kinase. That'll have the effect of inactivating glycogen phosphorylase and activating glycogen synthase. again, this makes sense. Insulin is released when glucose levels are high. And so you want to activate glycogen synthase to store the glucose, to lower blood glucose back to our dynamic steady state of 5 millimolar. And so you want to activate glycogen synthase and activate glycogen phosphorylase. So the insulin activates glycogen synthase and inactivates glycogen phosphorylase. Nothing special here, guys. And remember, insulin goes to that tyrosine kinase receptor. You got dimerization, cross phosphorylation, binding of effector protein that then go ahead and activate a bunch of different pathways. One of those pathways, this pathway here. Now we can move on then to gluconeogenesis. That's all I have to say about regulation of glycogen metabolism. It's not that complicated. So gluconeogenesis is the process by which the body creates glucose de novo, from nothing, or from relatively unrelated compounds. Gluconeogenesis begins in the mitochondrial matrix and ends in the cytosol, assuming you start with pyruvate. It can start in the cytosol, depending on what you start with. Gluconeogenesis is the reverse of glycolysis with a few new enzymes. So it's not exactly the reverse, but the reverse. At the irreversible steps of glycolysis. The gluconeogenic enzymes are only present altogether in total at the liver plus the kidney. So gluconeogenesis only occurs at the aforementioned organs. Okay. There are enzymes that are phosphoenolpyruvate carboxykinase, pyruvate carboxylase, and fructose bisphosphatase, and glucose-6-phosphatase. Gluconeogenesis is an energy-demanding process. It takes a lot of energy, an ATP, GTP, to run gluconeogenesis. It requires the use of 6 ATP equivalents, 4 ATP, 2 GTP, and 2 NADH molecules to convert 2 pyruvate molecules into 1 glucose. Remember, glucose gets cleaved in half to GAP and DHAP, so that's why we get 2 pyruvates out, and then we have to convert those 2 pyruvates back to then 1 glucose. Gluconeogenesis is primarily regulated by levels of fructose 2,6-bisphosphate, F26BP. Fructose 2,6-bisphosphate levels are ultimately determined by glucagon and insulin signaling and some other hormones to a lesser extent, as well as mass action effects. So let's go through it. Gluconeogenesis starts in the mitochondria and ends in the cytosol. Okay? Just orienting you. So again, gluconeogenesis is the reverse of glycolysis with a few new enzymes. There are sites here where the reaction is relatively irreversible. Yeah, they have a very large negative Gibbs free energy, which actually implies that they have some equilibrium value. But the reality of it is so negative that we say that it's irreversible. Okay, so we had this step here, this step here, this step here. So boom, boom, boom. We need to have other enzymes to catalyze that. The rest of it can go right through this because these are all reversible reactions. So the first step is to take pyruvate, convert it into oxaloacetate via pyruvate carboxylase. We know it's carboxylase, so it uses biotin cofactor, ATP, CO2. And then we convert oxaloacetate into phosphoenolpyruvate via phosphoenolpyruvate carboxykinase. Input GTP, output GDP, and CO2. Okay? Then we can go through all these little steps here until we get to fructose 1,6-bisphosphate. Where at that point, PFK, I told you the major regulatory site, and if they're reversible, we need another enzyme. So fructose bisphosphatase will convert that into fructose 6-phosphate, go through phosphoglucose reversible reaction to glucose 6-phosphate, and then glucose 6-phosphatase to form then glucose. These enzymes are only found in liver and kidney. So gluconeogenesis only occurs at the liver plus the kidney. Okay? And notice that, so how much ATP do we have to put in? Well, we have an ATP here, right here. We have a GTP right here, and we have an ATP right here. Of course, this says forming ATP, but really you have to do the reverse reaction, so we're going to break down the ATP. So to convert 2 pyruvate molecules back into 1 glucose molecule, it takes 1 plus 2 times 2, 6 ATP equivalents. Okay, 4 ATP, 2 GTP. Another thing to note is that you have to consume NADH here. You have to use up NADH here, so it takes 2 NADHs as well, because you have to use this NADH and do the reverse reaction to then get back to GAP and DHAP. So we're going to need some cytoplasmic NADH, and we can't get it from glycolysis because that would defeat the purpose. If we had to run glycolysis to get the NADH to run gluconeogenesis, we wouldn't be doing anything on a net basis. So we have to get the NADH from somewhere else. So let's go through it. Let's go through it one by one here. Not one by one because this is nothing new to you. Pyruvate, so it starts in the mitochondrial matrix. So the first little problem that we have is we need to get pyruvate out into the cytosol, and we need to do that And it's actually a little weird. It's a little weird because it has to go through an oxaloacetate intermediate within the mitochondrial matrix to then get out into the cytosol and form oxaloacetate again. So you take pyruvate carboxylase within the mitochondrial matrix. You have to use CO2, and remember that mechanism. It's just that biotin cofactor that had this amide group that can attack the alpha carbon here. I mean the carbonyl carbon of the CO2 molecule. Okay, and then put it onto the substrate, which is pyruvate. And it takes ATP to do that. It takes energy. You have to put input energy to do that. So you form oxaloacetate. That's a beta keto acid. Take malate dehydrogenase to form malate, and you have to use up an NADH to form NAD+. You take a malate carrier. Malate goes out. An inorganic phosphate comes in. Okay, we have malate into the cytosol now. And now malate dehydrogenase, we run that reaction except in the other direction. Form NADH. Now we have cytoplasmic NADH right there. We have oxaloacetate. And then we convert oxaloacetate via phospholipyruvate carboxykinase into phospholipyruvate. And we have to input a DTP to do that. I don't really care that you know the mechanism for this. I mean, it's just not worth going through. You already know the mechanism for this. It's just use that biotin cofactor, that nucleophilic attack that I showed you earlier. You get a nucleophilic attack onto the CO2, and then the pyruvate then does an aldol-like reaction, aldol condensation onto the CO2. So again, boom, boom, boom. Those are points that we have to go through. We went through this. We went through this to form phospholipyruvate. Now we just go phospholipyruvate, 2PG via enolase, 2PG via phosphoglycerate mutase at 3PG, phosphoglycerate kinase to 1,3-BPG, 1,3-BPG to via glyceraldehyde 3-phosphate dehydrogenase, use up an NADH, form GAP and DHAP. And then we via aldolase form fructose 1,6-bisphosphate. Then we use fructose bisphosphatase to form fructose 6-phosphate, fructose 6-phosphate via phosphoglucose from glucose 6-phosphate, glucose 6-phosphatase from glucose. Clearly, these are just hydrolysis reactions here. That's why I'm not mentioning mechanism. Okay, all this happens. So, the next step, at least what we need to talk about next, because obviously this isn't complicated, is what can feed into this pathway. Because where are we getting pyruvate from? We need to get it from somewhere. Well, pyruvate, amino acids, lactic acid, the glycerol, and all-chain fatty acids can contribute to gluconeogenesis. So right here, I wrote the one-letter code of the amino acid and where they feed into the Krebs cycle, citric acid cycle, TCA cycle. Okay. One thing to notice here is that acetyl-CoA cannot be converted back into glucose. Okay. You cannot. You can't do gluconeogenesis with acetyl-CoA. So any amino acids that feed into acetyl-CoA are solely ketogenic, unless they feed into some other pathway. So leucine, and lysine are solely ketogenic. Keep that in mind. That's one thing. If there's anything I want you to know about protein catabolism, it's that. Remember, glycerol can be converted into DHAP, right? And then go into gluconeogenesis that way. Fatty acids, however, are broken down into the even chain fatty acids can't contribute to gluconeogenesis because they form acetyl-CoA, and you don't go backwards to pyruvate from acetyl-CoA. And the odd chain fatty acids form propionic acid that can then be converted to a 6-8 through a series of reactions. And so the odd chain fatty acids can fit into right here. Some other things that I want to mention is why can't acetyl-CoA be used for gluconeogenesis? When I was taking biochem, I always thought, well, can't it just go through all this to get to, let's say, oxaloacetate and then, and then go into gluconeogenesis? Well, no. You know what? Because you had to use up a pyruvate to do that. So on a net basis, you're not gaining anything from that. If you use acetyl-CoA on a net basis, you're not really gaining anything from that. Acetyl-CoA did not contribute to gluconeogenesis. And there are a few more parts to it. So some other parts of it is that oxaloacetate, when you're doing gluconeogenesis, it's being siphoned off. And you know that citrate synthase uses an obligate binding mechanism, and oxaloacetate has to bind first. Well, if oxaloacetate is being siphoned off into gluconeogenesis, then you can't run the citric acid cycle to then reform oxaloacetate from acetyl-CoA. So that's another reason why it doesn't produce gluconeogenesis. And the other reason is that it goes into forming ketones, and it gets pulled into those reactions. Ketones. Let's put K as ketone synthesis. So that's why. I mentioned amino acids contributing. A lot of times the protein catabolism works via using transaminases that use the PLP cofactor to catalyze their reactions. Okay? And it goes through this reaction mechanism, but the reaction mechanism is not important. Appreciate the fact that transaminases transfer amine group. So you move this amine group from here to here. To here rather. And you move this double bond oxygen on this compound to this compound. All you do is transfer where you find the amine groups and where you find the double bond oxygen groups. And then these breakdown products often go into then TCA cycle, like aspartate plus alpha ketoglutarate form oxaloacetate plus glutamate. Okay, so there it is. Transaminases used in protein catabolism. So burden shifting cycles, the Glucose-Alanine cycle. So working muscle tissue, skeletal muscle, typically likes to run the glycolytic cycle, especially the fast muscle fibers. They typically want to run the glycolytic cycle because oftentimes when they have these fast burst movements that they have to do, they need ATP very quickly, and we know that we can upregulate the glycolytic cycle about 100x its normal rate. And so working skeletal muscle likes to run that cycle. And so a way to conserve ATP and to keep running that cycle is the glucose-alanine cycle. So why don't we start at liver with glucose? Glucose gets pushed out into bloodstream by the GLUT2 transporters at the liver and get to the skeletal muscle where they're uptaken by GLUT4 transporters. The glucose then can either go into glycogenolysis or glycolysis rather, and the glycogen goes to glycogenolysis to form pyruvate at the end of glycolysis, right? And we form 2 ATP and we get 2 NADH out of that. Now, the glucose-alanine cycle is all about conserving ATP at muscle. So what it's going to do is going to take the pyruvate that you made, and it's going to convert it into alanine via transaminase. So glutamate into alpha-ketoglutarate, and then pyruvate into alanine. Alanine is then going to go into bloodstream and get to liver, where it's going to be converted into pyruvate, gluconeogenesis, and into glucose. Now glucose can then travel back to skeletal muscle, where it can go through the glycolysis and form more ATP very quickly and go through the cycle again and again and again. It's a cycle. That conserves ATP at muscle, because now muscle does, it saves ATP at muscle, and it conserves the muscle time. It saves the muscle time because now it doesn't have to wait for it to go through the TCA cycle and then electron transport chain to generate ATP. No, we can convert it to alanine, get it to liver, and then liver convert it into glucose via gluconeogenesis, send it back to the skeletal muscle to then run glycolysis, which is the faster reaction. So it saves you muscle and time. ATP and time at the muscle. It's a glucose-alanine cycle. Okay, so why don't we move on to lactic acid, which can also contribute to gluconeogenesis. You know that when we're exercising, we often generate lactic acid or lactate, okay, deprotonated form. Lactic acid is produced when we run the glycolytic cycle, when we run glycolysis and then run the fermentation into lactate. And in doing so, we use up an NADH to form the NAD+, to then rerun glycolysis. That's why we do the glycolytic cycle, to run glycolysis over and over. Well, after we've run and we're tired and we finished running, we often have hypoglycemia. We have relatively low blood sugar. And so what we want to do is then run gluconeogenesis. So we want to run the reverse of this reaction and use up NADH, form NAD+, to form then pyruvate, to then convert pyruvate via pyruvate carboxylase into oxaloacetate, and then via phosphorine and pyruvate carboxykinase into phosphorine pyruvate to then go through gluconeogenesis. So that's how lactic acid can contribute. Another thing that I want to mention here is alcohol metabolism, okay? So alcohol can inhibit gluconeogenesis indirectly. So what happens is that ethanol, I'm talking about ethanol now, when you intake ethanol, okay, we need to then break down, we have to metabolize ethanol into something else. It's acetaldehyde. You know, the fact that I'm telling you it's an aldehyde, the ethanol has been oxidized, okay? It has been oxidized, so something had to be reduced. So the reducing equivalent that was made was NADH. So when we binge drink ethanol, okay, we're going to increase the amount of NADH we have, okay? That, by mass action, is going to increase the formation of lactate. And you know what? If you increase the formation of lactate, de facto then you're decreasing the formation of pyruvate. If you decrease the formation of pyruvate, you have less pyruvate being converted into oxaloacetate via pyruvate carboxylase, less oxaloacetate being converted into phosphatidylpyruvate via phosphatidylpyruvate carboxykinase, you have less gluconeogenesis. And that's a big problem for liver, because liver is keenly involved in regulating blood glucose levels, and if it cannot run gluconeogenesis effectively at the rate at which it wants to, you're going to become hypoglycemic. Okay, you're going to become hypoglycemic. So alcohol intake can cause hypoglycemia. Another burden shifting cycle, the Cori cycle. It's very similar to the Glucose-Alanine cycle. It's all oriented in saving energy and saving time at muscle. So why don't we start at liver? You take glucose from the liver, okay, and it sends it out into the bloodstream, and that's the skeletal muscle. Again, it can be converted into glycogen or used up in glycogen and glycolysis to form lactate. The glycolytic cycle, okay, or glycogenolysis to form lactate. Now, you know that we need NAD plus to keep running glycolysis. And the way we get that is converting it into lactate. And that allows us to then rerun glycolysis, okay? But we also need glucose to run glycolysis. So instead of skeletal muscle running gluconeogenesis, which it can't anyway, but even if it could, it wouldn't, it sends a lactate to liver to run gluconeogenesis, okay, to form glucose, to then send it back to skeletal muscle to run glycolysis. So it saves ATP at muscle, and it shifts the metabolic burden from working muscle to liver. Pretty sensical. Note the trend. Both the Glucose-Alanine cycle and the Cori cycle are bent on saving ATP at muscle tissue during times of exercise. The Glucose-Alanine is more about saving time than ATP, but both save you ATP and metabolic burden. A broad stroke, again, take pyruvate via pyruvate carboxylase using ATP and ATP plus CO2 to form oxaloacetate. And then oxaloacetate via phosphatidylpyruvate carboxykinase to form phosphatidylpyruvate. You got to use the GTP. Then you just go through glycolysis, use up an ATP here, and use up an NADH here, gap DHAP, and then you finally get to fructose 1,6-bisphosphate via fructose bisphosphatase to form fructose 6-phosphate. And then fructose 6-phosphate would be a PGI, phosphoglucose glucose 6-phosphate, and then glucose 6-phosphatase to form glucose. Again, this only happened at liver and kidney, okay, and it takes up 6 ATP equivalents. Energy demanding process. So we can move on then to the regulation of gluconeogenesis. So it's reciprocal regulation, just like it's reciprocal regulation of most things, okay? We're trying to maintain a dynamic steady state, so if you want to do that, you better have reciprocal regulation. Again, these are the steps, and these are the different enzymes involved. Why don't we work through PFK? So PFK, phosphofructokinase, I told you, is the major regulating point. Fructose 2,6-bisphosphate will activate it. We haven't talked about that yet. I'll talk about it soon. AMP and ADP will activate. ATP will inhibit. That makes sense. AMP, the major regulator in terms of comparing ATP and AMP. AMP just indicates low levels of energy, so of course you want to run glycolysis. Whereas if you had high levels of ATP, of course it's going to inhibit. If you want to talk about pyruvate kinase, ATP will inhibit, and fructose bisphosphate will activate via a mass action effect. Why don't we talk about the gluconeogenic hormones now? Fructose bisphosphate will be inhibited by fructose 2,6-bisphosphate and AMP. Again, that makes sense. And pyruvate carboxylase will be activated by acetyl-CoA and inactivated by ADP, and PepCK will be inactivated by ADP. Now, acetyl-CoA is actually a required cofactor for pyruvate carboxylase. It is an allosteric activator that is not a cofactor, but it's an allosteric activator that is required for pyruvate carboxylase function. Required. Okay? Now, why don't we talk about fructose 2,6-bisphosphate? So, when I went through glycolysis, we talked about PFK, phosphofructokinase. Well, this is the isozyme of PFK. It's PFK2. While PFK catalyzes the formation of fructose 1,6-bisphosphate, PFK2 activates the formation of fructose 2,6-bisphosphate. Clearly, that's not a glycogenic substrate, but it is a substrate that acts as an activator, a product rather, that acts as an activator to PFK, regular PFK, PFK1. PFK1 is the regular PFK. PFK is found in glycolysis. Now, it makes sense that fructose 2,6-bisphosphate activates PFK. Why? Well, think about it on a mass action effect here. Mass action effect. If you increase the amount of fructose 6-phosphate and you increase that via having a lot of glucose-6-phosphate or having a lot of glucose, by mass action, you're going to increase the formation of fructose 2,6-bisphosphate. If you increase the formation of 2-fructose 2,6-bisphosphate, that just means you have a lot of glucose. So, of course, this is going to act as an activator for PFK or glycolysis. Makes complete sense. Now, another thing to notice and to realize here that fructose 2,6-bisphosphatase is actually just another part of the PFK enzyme. It is the other side of it. So it had a kind. So PFK and fructose bisphosphate is part of the same enzyme, same protein. They have two different active sites. They have a kinase domain and they have a phosphatase domain. And these two different domains catalyze the different reactions that we see. So it's a bifunctional enzyme. Okay? Not complicated. Again, so that's why this would activate PFK, because it makes sense. Now, there are different hormones that can regulate the level of fructose 2,6-bisphosphate, and hence regulate gluconeogenesis versus glycolysis. Why don't we deal with glucagon first? If you decrease the level of glucose is gone, glucagon secretion increases by the alpha cell of the pancreas. Again, it binds to the G protein coupled receptor, called the conformational change in the G protein such that G alpha releases GTP and binds GTP, causing a dissociation from G alpha and G beta gamma. G alpha activates the adenylyl cyclase, converted to cyclic AMP. Cyclic AMP activates protein kinase A. Protein kinase A called this for then the phosphorylation and the inhibition of PFK2 and the activation of fructose bisphosphatase. What did that do then? That'll decrease fructose 2,6-bisphosphate concentrations, and that will increase gluconeogenesis and decrease glycolysis. Not complicated. That all makes sense. You don't even have to memorize this because that just makes sense. How about we increase glucose levels? Well, that'll increase insulin secretion by the beta cells of the pancreas. That'll increase the activation of phosphoprotein phosphatase 1. That'll cause for dephosphorylation of PFK2 and fructose bisphosphatase, and the activation of PFK2 and the inhibition of fructose bisphosphatase. Of course, that's going to increase concentration of fructose 2,6-bisphosphate. That's going to decrease gluconeogenesis and increase glycolysis. That all just makes sense. Not complicated. Now, one complication, if you would, that would arise here is that in the heart tissue, right, it's actually the opposite effects occur for glucagon and insulin signaling. So for glucagon signaling, glycolysis will actually increase at the heart, okay? Whereas for insulin signaling, glycolysis, probably not changing. But in the heart, the opposite effect is that glycolysis increases when you increase glucagon signaling. When you overview gluconeogenesis. Gluconeogenesis is the process by which the body creates glucose de novo from nothing or from relatively unrelated compounds. Gluconeogenesis begins in the mitochondrial matrix and ends in the cytosol, assuming you start with pyruvate. Gluconeogenesis is the reverse of glycolysis with a few new enzymes at the irreversible steps of glycolysis. The gluconeogenic enzymes are only present altogether at the liver plus kidney, so gluconeogenesis only occurs at the aforementioned organs. Gluconeogenesis is an energy-demanding process. It takes about 6 ATP equivalents and 2 NADH molecules to convert 2 pyruvates into 1 glucose molecule. Gluconeogenesis is primarily regulated by level of fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate levels are ultimately determined by glucagon and insulin signaling and some other hormone, then to a lesser extent, as well as mass action. Yeah, okay, other hormones like cortisol, but let's not get complicated. You know, I'll mention it, but we won't get too complicated with it. We'll do integration now, a little bit of integration. We already integrated a lot, but again, so we'll look at insulin and glucagon effects at the systemic level. There's nothing new. Increased blood glucose levels. Beta cells increase release of insulin. Through a tyrosine kinase receptor, protein kinase B phosphodiesterase pathway at the fat, you increase glucose uptake, you increase fatty acid synthesis, and you decrease beta oxidation. At liver, you increase glucose uptake, increase glycolysis, increase glycogenesis, increase fatty acid synthesis, decrease gluconeogenesis, decrease beta oxidation. Increase glucose uptake at just general cells and muscle, you increase glucose uptake, increase glycolysis, increase glycogenesis. And blood glucose levels lower. If you decrease blood glucose levels, you alpha cell of the pancreas release glucagon. Through a G protein coupled receptor cyclic AMPK pathway, you increase gluconeogenesis, increase beta oxidation, increase glycogenolysis, decrease glycolysis, and decrease glycogenesis. And at fat, you increase beta oxidation, decrease fatty acids, all directed at returning blood glucose towards set point, which is, again, the dynamic steady state that we're maintaining. Maintaining the dynamic steady state. Primarily, that is coordinated by the pancreas and the liver. Okay, why don't we go into molecular detail? So regulation of fat metabolism. Epinephrine or glucagon binding to G-protein coupled receptors will go through this adenylate cyclase pathway, activating cyclic AMP. Cyclic AMP will activate PKA. PKA will phosphorylate ABDH5 as well as paralipin, which was sequestering ABDH5. That will release the sequestering effect and allow ABDH5 to activate adipocyte triacylglycerol lipase to convert TAGs into DAGs, releasing free fatty acids, DAGs via hormone-sensitive lipase into MAGs, monoacylglycerol lipase to form free fatty acid and glycerol. You know that free fatty acids will then go through beta oxidation to form acetyl-CoA and power then gluconeogenesis. Okay, and glycerol will then be converted into DHAP, which then can get converted into then glucose. Now, if insulin binds to their receptor, okay, it'll essentially go through this rather complicated pathway to form a phosphodiesterase at the end of it, to then inhibit cyclic AMP to prevent all these downstream reactions from occurring. Okay, so that's regulation of fat metabolism within the adipocytes. So that's what's happening there. I also mentioned last time the hormone cortisol. So physical emotional stress will cause for the release of cortisol release of CRH from the hypothalamus, which will go into the median eminence through that portal circulation into the anterior pituitary and bind to the cell there, and cause for then the release of adrenal corticotrophic hormone, which then calls for the release of cortisol from the adrenal gland, adrenal cortex. It's a steroid hormone. Okay, it's a glucocorticoid. First clue. It's a gluconeogenic hormone, and it's directed at, for immune cells, suppressing a response, muscle protein catabolism to then contribute to gluconeogenesis, liver gluconeogenesis, adipose tissue lipolysis to contribute ATP to run gluconeogenesis, and increase appetite at the hypothalamus to start eating, stress eating, if you would. It's a gluconeogenic hormone, guys. Again, we already went through glycogen metabolism. We said that kinase signaling leads to the activation of glycogen phosphorylase and the inactivation of glycogen synthase, because glucagon activates kinase signaling and glucose is gone at the mnemonic. So, of course, we want to increase blood glucose levels by doing so. And insulin activates phosphatase signaling that inactivates all the things that glycogen would have activated. So, insulin will cause for the activation of glycogen synthase and inactivation of glycogen phosphorylase. And remember, this regulation of cellular respiration is primarily determined by reduced cytochrome C levels, which is determined by all these little allosteric effectors here. But the main one to remember is PFK, because that is the major flux controlling enzyme of glycolysis. And we've added one more thing, which is fructose 2,6-bisphosphate. It's levels of ATP and NADH that regulate these things. But PFK, the major flux controlling enzyme, which controls the level of the reduced cytochrome C that would then oxidize complex 4 to then reduce the oxygen into water and form then that proton concentration gradient that can then be dissipated by the ATP synthase to form ATP. That is controlled by reduced cytochrome C levels, which is controlled by the allosteric effector, the mass action effects here. Insulin increases glycolysis, glucagon increases gluconeogenesis. Again, we went through functions of AMPK last time, so it's worth mentioning. AMPK working at heart will increase glycolysis. That's a reminder that heart acts a little bit different. You never want to slow down metabolism at heart. You don't want your heart to stop. At liver, AMPK, again, AMPK will be active when there's high levels of AMPK, when there's high levels of AMP. When there are high levels of AMP, when we have no energy. So you're going to inhibit gluconeogenesis at liver. You're going to inhibit cholesterol synthesis at liver. You're going to inhibit fatty acid synthesis at liver. Okay, and you're going to increase beta oxidation to form ATP at liver. At muscle, you're going to inhibit glucose being converted into glycogen, so you're going to inhibit glycogen synthase, and you're going to activate the conversion of glucose into pyruvate via glycolysis. And you're going to activate the citric acid cycle electron transport chain. You're going to activate beta oxidation at muscle. You're going to and one thing that is weird here about AMPK at this adipose tissue is that AMPK is actually going to inhibit triacylglycerol being converted into fatty acids, free fatty acids, to then form beta oxidation. And the reason for that is thought that it's counterintuitive, but the rationalization at least, is that you don't want to waste energy by breaking them down to triacylglycerol to then reform them later, because we're already running all these pathways. That's the rationalization. Again, AMPK, the big regulator too. So at AMPK at the systemic level will inhibit fatty acid synthesis and inhibit lipolysis. It'll inhibit fatty acid synthesis and cholesterol synthesis at liver, inhibit insulin secretion at the pancreas, And it'll activate glucose uptake, fatty acid oxidation, glycolysis at heart, and activate glucose uptake and fatty oxidation at muscle. On a molecular level, it'll activate PFK2 at heart, which will increase cardiac glycolysis. Because of the fact that you activate PFK2, you increase fructose bisphosphatase, you're going to increase cardiac glycolysis. It'll increase the secretion of GLUT1 and GLUT4 transporters into the membrane, increasing glucose uptake. It'll inhibit eukaryotic elongation factor 2 or mTOR and inhibit translation. It'll inhibit glycogen synthase. It'll inhibit GPAT, which will inhibit triglyceride synthesis at adipose tissue. It'll inhibit HMG reductase, HMG-CoA reductase, which will inhibit isoprenoid synthesis, also inhibit ketone body synthesis. And it'll inhibit hormone-sensitive lipase, which will inhibit lipolysis. And it'll inhibit acetyl-CoA carboxylase and fatty acid synthase to activate fatty acid oxidation and inhibit fatty acid synthesis. So the last thing we have to cover then is starvation. Starvation is a severe deficiency in energy intake needed to maintain an organism's life. You must maintain about 2.5 millimolar glucose level to maintain consciousness. Glycogen stores are depleted in roughly 60 hours during starvation conditions, and the liver undergoes gluconeogenesis and produces ketone bodies. Fat undergoes lipolysis and beta oxidation to power gluconeogenesis, and muscle undergoes protein catabolism to contribute substrates to gluconeogenesis. Here is a list of our different calories that we have in terms of kcals, okay, kcals, big C, calories, of the different fuel metabolism, fuel reserves that we have. Fat, we have a whole ton. You'll notice that fat is the big source. That is the big source of fuel that we have. The rest of it is not that high. You'll notice that we have so much fat that we can last up to 3 months while starving, but we'll typically die before then for things like dehydration. But if you're really lucky, you have all the water in the world, then you may survive for about 3 months on your fat stores for a normal 70 kilogram man. Okay? Some other things I want to impress upon you. During starvation, we shift metabolism from glucose to ketone bodies. So you'll notice at about day 2, we start producing ketones. And you go into what's called ketosis. Okay? Yeah, it's kind of dangerous because you become acidotic, but it helps us in the fact that our brain can then rely less upon glucose and more upon ketone bodies because we're starving. We don't have a lot of glucose, but we have a lot of fat that we can convert into acetyl-CoA and then convert into ketone bodies. All right? So we have a shift in metabolism from glucose to ketone bodies during starvation. And just to impress upon you about that, on day 3, the fuel used by glucose by brain is about 100 grams of glucose per day. Whereas ketone body is about 50 grams per day. Well, at day 40, you're using about 40 grams per day of glucose at brain and about 100 grams of ketone bodies per day by the brain. Okay? And that's pretty much it about starvation. You last about 3 months. You need water, otherwise you'll die in a few days. And you have a shift in metabolism from glucose to ketone bodies. It can become dangerous. 1, because, you're not eating much. But 2, because your blood can become acidotic from the ketone bodies, the formation of these acidic ketones, and you can die because your enzymes are no longer functioning at the proper pH. So that's starvation, that's metabolism. I hope you enjoyed the lecture.